Hello, good morning or good afternoon everyone, depending on where you are joining us from, and welcome to today's webinar. My name is Annabelle Chaplin from Business Review Webinars, and I'll be your host. It's our pleasure to have BRIM Research, a division of Capsugel Dosage Form Solutions, with us today, who will be discussing the fundamentals of hot melt extrusion. Today's guest speakers are Charlie McLaughlin, who is an Engineering Technical Group Lead at BRIM Research, and Scott Bone, who is a Chemistry Technical Group Lead, also at BRIM Research. I'd like to welcome you to our webinar platform On24. You will notice that this webinar is browser-based, so if you disconnect, please click the link that you received via email to rejoin the session. All of the icons along the bottom of your screen are the interactive widgets that we have on offer today, so please interact with them all throughout the session. You can send in questions by using the Q&A widget or by using the questions box in the top left-hand corner. We will try and get through as many of your questions as possible. However, if we do not have time to answer them all in this webinar, then Charlie and Scott will get back to you at a later date. Please use the help widget if you experience any technical difficulties or require more assistance. So, without further ado, please allow me to welcome Charlie and Scott.
Thank you, Annabelle. In the next 45 minutes, we will be presenting the fundamentals of hot melt extrusion. First, I would like to give a brief overview of Dosage Form Solutions and BRIM Research. Dosage Form Solutions and BRIM Research utilize an integrated product development approach, ensuring one partner for formulation development, process development, and production of innovative intermediate and solid dosage form and final solid dosage forms. Living our values, project focuses center on innovation, quality, and customer service to ensure we are doing our best to meet client and patient needs. For a specialized set of technologies, we have the ability to offer formulation development, process development, and scale up to commercial manufacturing. We bring deep science and engineering capability that complements deep drug development capabilities that our customers are bringing to the table. Our breadth of technology and expertise allow us to develop an understanding of the optimal drug technology for a given product and offer unbiased recommendation for formulation approach. Our integrated approach to problem statement allows us to increase the speed and decrease the risk associated with formulation and process transfers. Included on this slide is an overview of our technology platforms and offerings. Bioavailability-Enhancement will be the focus today, with a focus on hot melt extrusions. SDD, lipid-based formulations, nanotechnologies, and particle size reduction are also technologies at Dosage Form Solutions and BRIM Research to be applied for bioavailability enhancement. Functional capsules, multi-particulate formulations, osmotic technologies, abuse deterrent formulations, and high-potency handlings are just a few examples of our additional technologies. These technologies and many others make up our multifaceted technology platform approach. Our breadth and depth and range of offerings as well as fundamental science approach are utilized for technology application and formulation selection. Now we will proceed with our talk on hot melt extrusion fundamentals. So I'd like to start with a little context. Depicted on the slide here are the DCS classification systems. Today, we will be focusing on the DCS Class II limited bioavailability by solubility and dissolution rate. The slide here depicts percentages of marketed products as well as percentages of candidates. Discovery and development portfolios across the industry are resulting in more compounds in the DCS Class II. BRIM Research and Dosage Form Solutions have many technologies to address this problem, and today we will focus on hot melt extrusions. Technology selection is an important part of formulation. Bioavailability enhancement can be achieved using many technologies, and this map is a representation of how those technologies can be differentiated. Based on our experience, crystalline salts, amorphous formulations, and lipid-based technologies are the most widely applicable. However, there is a very large overlap in these technologies. Typically, it is a dimension not shown on this plot that guides formulation selection or technology selection. Examples of these would be target PK profiles, metabolism, permeation, or dose limitations. One solution to bioavailability enhancement is amorphous solid dispersions. These dispersions are non-equilibrium forms of the material. They are high energy and can generally be stabilized using kinetic or thermodynamic stabilization. All topics we will be covering today. There are generally two ways to manufacture an amorphous solid dispersion, those being hot melt extrusion as well as spray-dried dispersions. Solid dispersions by hot melt extrusion are generally a subset of dispersions possibly manufactured by spray-dried. Hot melt extrusion is a continuous process that has low capital and low manufacturing costs. However, the formulations are limited to miscible systems at processing temperatures and require active components that are thermally stable. One consideration when comparing the two manufacturing processes for amorphous solid dispersions is the particle properties of the resulting powder. HME solid dispersions tend to have excellent flow and are generally amenable to direct compression. Spray-dried dispersion formulations tend to have good powder compressibility, but often require dry granulation in order for downstream processing. HME formulations have precedence in the market and have a good and long history. Within BIN research, we have worked on and have one marketed product with Pfizer, that being Zithromax. This formulation was a melt congeal, which takes advantage of an HME processing step in the manufacture of the formulation. As I mentioned, HME is well understood and a mature technology. However, it is being used more for solid dispersion technology than it has in the past and is an important tool for us to use in formulation selection. I will now pass it over to Charlie for an overview of the processing.
Thank you, Scott. Here on slide 8, we have a general HME process train for the amorphous solid dispersion manufacturing. Generally with this process, the first step is going to be a physical blending. For a binary system of the active pharmaceutical drug and a polymer, we will generally process this physical blend first. Once the blend is completed, we will proceed with feeding the extruder, generally with a gravimetric or a volumetric feeder. Once the material is fed, it processes through the actual extruder and undergoes the hot melt extrusion process. We will further go into the technical details of the extrusion process later on, but once the extrusion is complete, the material passes next into a cooling apparatus, which takes the melted material back down to a cooler temperature and processes it into a pelletizing piece of equipment to further reduce the particle size. The last process that we will discuss is a milling process that takes that extruded material and reduces it to a smaller particle size, more amenable towards a downstream process, whether or not that is an immediate release tablet or various other dosage forms. So at the heart of this process is the extruder itself. And so we will go ahead and discuss some of the variables that are used in the extrusion process. Bend Research prefers to use twin screw co-rotating fully intermeshing extruders. The extruder itself has various different variables that we are allowed to take a look at during the process optimization and defining the process operating space. The twin screw co-rotating fully intermeshing extruder is a preferred piece of equipment for solid dispersion processing. The twin screw co-rotating fully intermeshing extruder has better mixing than a single or non-intermeshing extruder. The twin screw extruder consists of two co-rotating screws in a segmented barrel that has different cooling zones within each, different cooling or heating zones within each segmented barrel. Some of the independent continuous process variables that we can take a look at, and by continuous I mean that they can be changed as the process is being manufactured. These variables are screw speed, feed rate, and barrel temperature. Some of the independent non-continuous process variables that we can take a look at are screw design, barrel design, and die design. And then some of the dependent variables that we take a look at are melt temperature, residence time, downstream processability, and dispersion quality. So the unit operation and the screw design for the manufacture of amorphous solid dispersions is shown here. This is a typical screw design for a targeted process that we have. And then within the extrusion process itself, we typically like to say that there are 5 different unit ops that occur within the barrel and as the material processes down through the intermeshing screws. The first unit op that we are going to discuss is the feed section. The feeding takes place at the first part of the screw. The polymer and API can be introduced independently or as a blend, either volumetric or gravimetrically. This powder is fed into the extruder via the feed throat. The feed barrel is actively cooled to prevent low melting point materials from sticking. The next unit op that we will discuss is the conveying and melting zone of the extruder. This occurs after the feed zone, and the primary purpose of this unit op is to move powder away from the feed zone. Energy is first introduced via heat transfer from the blocks and shear introduced via the screws. The energy requirement and the screw configuration is often dependent upon the melting point of the components and the particle size of the components that are being introduced. The next unit op that we will talk about is the mixing. The goal of this process is to provide enough mixing to produce a well-mixed material at the molecular level or within the single phase. The amount of mixing depends on whether or not we are dissolving the API into the polymer or melting the API into the polymer matrix. The two main types of mixing that we will be discussing are distributive and dispersive. The first type of mixing that we discuss is dispersive mixing, which is accomplished with kneading blocks. The kneading blocks in a co-rotating twin screw extruder often offer a versatile force combination of shearing, elongating, kneading, and stirring actions. You can specify the type of kneading block that you would like to use based upon the amount of energy you want to introduce into the system. The larger the angle provided on the kneading block, the more dispersive mixing. So a specified 90-degree kneading block will have more dispersive mixing when compared to a 30-degree kneading block, which will have more conveying mixing within that kneading block. The next type of mixing that we will discuss is distributive mixing. This is accomplished with combing elements and small angle kneading blocks. You can see in the graphic in the middle of the slide that the very top circle in that pyramid is the starting material of the API and the polymer, and for the dispersive mixing on the left, we are taking those particles and breaking them down and mixing them at the molecular level. On the right side is the distributive mixing, where you're taking those two elements and fully distributing them throughout the amorphous dispersion. And then obviously at the bottom circle is the end goal of that homogeneous amorphous dispersion. So generally for the HME process, we proceed with distributive mixing followed by more dispersive mixing. And then for a screw design, mixing intensity is a function of the residence time, specifically the span of residence time, and screw speed. So faster screw speed allows for better mixing, and slow speed allows for slightly less mixing. The next unit op that we'll take a look at is venting. The primary purpose of venting is to remove residual moisture and gas or air entrained within these powders. If necessary, a vacuum can be used to help pull a vacuum on top of the extruder to help allow for more venting of the material. The last unit op that we'll take a look at is the actual extruding of the material through a die. The final elements in the screw design are pumping screw elements that are used to extrude the solid dispersion out of a shaped die. The next slide here provides examples of the equipment that we have at Bend Research that we actively use for our development and GMP scale manufacturing of hot melt extrusions. The smallest extruder that we have is the 16 millimeter Eurolab Thermo Fisher Scientific extruder. This is our smallest extruder, and generally we like to use API quantities of 10 grams up to about 100 grams of material. The next development scale piece of equipment that we have is the 18 millimeter Lystritz twin screw extruder. We have both a GMP and a development unit for this piece of equipment, which allows for a seamless tech transfer and ensures that the development process closely matches the GMP process because of these same pieces of equipment. The largest extruder that we have here in-house is the 27 millimeter Lystritz extruder. Again, we have both a development unit and a GMP unit, which allows for seamless tech transfer processes. We have a variety of techniques that we can use PAT on during the extrusion process. We have Raman and NIR, which we routinely use to monitor our processes. In the middle picture on this slide, you can see the 18 millimeter Lystritz with the Raman probe attached to the die of the extruder to allow for PAT. You can see in the upper right corner we have a variety of dies that we can use for the extrusion process. These custom dies can be manufactured depending upon the need and application of the project. This next slide shows a variety of the ancillary and milling equipment that we use for the hot melt extrusion process. The first piece of equipment in the upper left is the high capacity steer chilled roll and attached flaker. Once the extruded material passes out of the die, the next process is to run it through this steer chilled roll. The steer chilled roll has high surface area and allows for rapid heat transfer to cool the material down to a glass-like substance, which enables the milling operation to become feasible. So once the extrudate is chilled, we want to reduce the native particle size of the amorphous dispersion. We have various pieces of equipment that we can use to reduce native particle size to prepare for downstream processing of the extrudate. The first piece of equipment that we have is a Fitz mill, which is a type of hammer mill. And here at Bend Research, we have an M5A and an L1A with a variety of screens to ensure that target particle size is achievable. Another form of milling equipment that we have is a co-mill, and we have a variety of sizes of co-mills which allow for a variety of throughputs. And with each of these co-mills, we have a variety of screens and impellers that allow us, again, to reach a target particle size that allows us to achieve a downstream processable material. In the upper right-hand corner, we have a picture of our K-Tron loss and weight powder and liquid feeders. These equipment are used for feeding the extruders both gravimetrically and volumetrically.
For all amorphous dispersions by hot melt extrusion, it's important to balance performance, manufacturability, and stability. We like to take these considerations and divide them up into the processing and the formulation. A successful amorphous solid dispersion is a function of both formulation, equipment configuration, and processing parameters that then result in a formulation with acceptable performance, manufacturability, as well as stability. Keys for these processes are to understand the physics within the extruder, as well as using models and experiments to understand both the process and the performance of the formulation. Presented on this slide is a formulation flow chart for an amorphous solid dispersion by hot melt extrusion. On the left-hand side of the slide in the center is where we begin with the physicochemical properties of the compound. Once the physicochemical properties of the compound have been evaluated and hot melt extrusion is determined to be the appropriate technology, we move on to the formulation selection and operating space. This will be the next topic that I will cover. It's important to ask questions when doing formulation selection and evaluating hot melt extrusion. One of the first questions we like to ask is if the drug has solubility in the polymer. A useful tool for guiding the formulation selection for thermodynamically and kinetically stabilized dispersions is the Flory-Huggins theory. This theory is the same model used to predict similar solvent and polymer chemistry. There are many assumptions that go into this theory, but it's very practical for our use in HME solid amorphous dispersions. This allows us to optimize drug loading as a function of processing temperature. The theory predicts three regions of an amorphous dispersion. For all dispersions at some given fraction of drug in the dispersion, the theory predicts that there will be a thermodynamically stable regime. This usually is limited by the loading of the drug in the formulation, and that is usually a very low loading. For each formulation, it can vary. However, limitations are low loading in the final dosage form, as well as slow quench rates for the formulation. Kinetically stabilized formulations fall under two regions, metastable as well as unstable. For the unstable region, because the delta G of mixing is greater than 0, these formulations will spontaneously phase separate. Where we like to formulate, or where we formulate the majority of our formulations, is in the metastable region. In this region, the delta G of mixing is less than 0, therefore phase separation is not spontaneous. However, the slope of the curve is positive, and phase separation, given infinite time, will occur. A second question to ask when evaluating hot melt extrusion is if the solid dispersion will have acceptable physical stability at storage conditions. As mentioned previously, there are three physical situations for amorphous solid dispersions. The first situation is a thermodynamically stabilized system where at infinite time the formulation will remain amorphous. We've included on this slide a few characterization techniques for determining which physical situation is present for your formulation. PXRD and modulated differential scanning calorimetry are the two techniques discussed on this slide. For a thermodynamically stabilized system, at infinite time, PXRD will remain amorphous, and DSC will have a single TG characteristic of a single phase amorphous dispersion. For the kinetically stabilized physical situations, there are two forms of phase separation. The first form would be phase separation into amorphous domains, and at infinite time, those two domains would be drug and polymer. So PXRD would result, once again, in an amorphous halo, while DSC would show two TGs, one of those characteristic of the polymer and one of them characteristic of the drug. The final physical situation for a solid amorphous dispersion at infinite time would be phase separation into crystalline domains. The PXRD for this characterization would be amorphous halo with API diffraction peaks, and the DSC would be a polymer TG representative of the polymer, and a drug melt representative of the neat API. So completely phase-separated API into crystalline domains. One tool that this slide depicts stability of STDs after 6 to 13 weeks. The x-axis on this curve is Tg minus T, which is a relative measure of drug mobility in the dispersion. As mentioned on the previous slides, kinetically stabilized formulations do have the propensity to phase separate, and one way to mitigate the phase separation is to store that formulation well below the glass transition temperature, limiting the mobility in the system. Shown on this slide is data collected on a number of compounds, all stored at relative conditions to the Tg. Shown on this slide is the data suggesting that 147 compounds were stable when stored at least 20 degrees Celsius above the Tg. There are some assumptions that go into this data that make this a conservative estimate, and those assumptions would include homogeneity of the ingoing material. At Bend Research, we utilize the rule of thumb that storing a formulation at least 20 degrees Celsius below the Tg will result in good predicted physical stability. Another tool that we can use for formulation selection are the Gordon-Taylor and Fox equation. These equations can be used to relate various polymers based on the glass transition temperature and effect of relative humidity. Shown on this slide is a prediction of various polymers for a compound with a glass transition temperature of 30 degrees Celsius at 0% RH. Following the rule of thumb provided on the previous slide, polymer selection can be used to help guide, the Fox equation and Gordon-Taylor equations can be used to help guide polymer selection. Another consideration for polymer selection is hygroscopicity. And presented on the slide is water vapor absorption data for various polymers. Compared here are HPMCAS, PVP, and Oedergit L100. As you can see, PVP has a very high water absorption relative to Oedergit and HPMCAS. And this can have a significant impact on predicted physical stability, or physical stability. With water content in a amorphous dispersion increasing, the plasticizing effects of the water will decrease the Tg and have a negative impact on physical stability. Another depiction of physical stability for various polymers is presented using contour plots. PVP K30 versus HPMCAS are presented as a function of glass transition temperature. The dotted line with the Tg of equal to 60 degrees Celsius is representative of a threshold where everything under the line is predicted to have good physical stability and everything over the line is predicted to have poor physical stability. Also included in the contour plots are the color related to the predicted glass transition temperature of the dispersion. As you can see, HPMCAS presents a much broader operating space with respect to predicted glass transition temperature as well as drug loading when compared to PVP K30. In addition to physical stability, chemical stability is also a consideration. This is typically monitored for formulations using rapid screening methods at accelerated conditions, for example, 50 degrees Celsius and 75% RH for very short time periods. The final question for consideration today is if the polymer enhances and sustains the solubility of the API in order to meet the target PK profile. One of the tools that we use to evaluate performance of formulations is an in vitro microcentrifuge dissolution test. This test is used to measure the solubility and sustainability of formulations. As presented on this slide, we use this to rank order formulations, both for active loading in the formulation as well as various polymers. Included in this evaluation are HPMCAS at 2 different grades, as well as HPMCP. The high grade of HPMCAS, or the H grade, is evaluated at multiple drug loadings. For a formulation with a given problem statement, this data can be used to help guide formulation selection. As presented here, all formulations exhibit a supersaturated solubility relative to the crystalline drug. However, some formulations have much better sustainability than others. For instance, the HPMCP has a very high supersaturated solubility. However, it does not sustain that supersaturated solubility for longer than 4 minutes. It then returns to a lower energy crystalline solubility. The HPMCAS-M has a supersaturated solubility and is able to sustain that solubility through 20 minutes. However, it does eventually crash back down to a lower energy solubility. Finally, HPMCAS-H, in addition to the other polymers, reaches a supersaturated solubility, but also sustains that solubility through 90 minutes. For this formulation, multiple drug loadings were also evaluated and shown to be relatively equivalent. While there is a difference in the dissolution rate for these 2 formulations, it is relatively small and the sustainability is very similar. And I apologize, the 15% drug load is depicted in the graph as the orange curve.
So the next discussion we will have in the formulation and process development flowchart is the definition of the HME processing operating space. So once the formulation operating space has been mapped out, defining the HME process operating space can be explored. And so the goal of this is to obtain a single-phase amorphous dispersion and know the variables that are required in order to reach that goal. So by defining the process space, we will also determine whether or not any additional processing aids are needed. Oftentimes, plasticizers are required in order to lower the melting point of the dispersion in order to help with physical and chemical stability. In other cases, it is often seen that various materials are also needed to help aid in the dissolution of the dispersion. We will also determine the processing constraints based upon the degradation seen of the API and/or the polymer. So on this plot, we have an example of an operating space for a hot melt extrusion process. This plot tries to quantify an operating space where a homogeneous solid dispersion with acceptable or no degradation of the API or polymer is obtained. The process of defining this space is an iterative process throughout the development and scale-up of the project. This particular plot assumes a fixed length over diameter and a screw design of the extruder. The first thing that we start with is by defining the top dashed line on the plot, and this line is the upper limit of temperature where no polymer or API damage is seen. The API degradation that we often see is a function of temperature, and so by maintaining a temperature below this line, it helps ensure that the API will have no chemical or physical degradation. This upper line also ensures that the API polymer temperature gradient does not affect the quality of the dispersion. The pictures on the bottom of this slide show a temperature gradient with a sample of each of the polymers, a PVP VA64 and an HPMCAS. And so this experiment allows us to look at the browning of the polymer versus temperature over an extended period of time. By running this experiment, we are allowed to look at whether or not we will have browning issues with the polymer as it passes through the extrusion, as it passes through the extruder. So the next line that we will look at is the complete dispersion line that bisects the upper temperature limit. This line defines the lower temperature at which we can achieve a complete dispersion. We will need to stay above this temperature line in order to ensure that we achieve an amorphous solid dispersion. The lines on the side of the mean residence time are the equipment operating lines. This allows us to take a look at the lowest feed rate and the fastest feed rate that ensures that our mean residence time stays within these limits and ensures that we stay within our operating space. The way that we generally like to measure the mean residence time is by using a tracer and various forms of PAT to help measure the mean residence time. So we can define the mean residence time when 50% of this tracer has passed through the extruder. Generally, we like to use approximately 250 milligram pulse of titanium dioxide passed through the extruder to monitor using Raman spectrophotometry. This data allows us to calculate or measure our mean residence time distribution. An example of this tracer plot is in the upper left. The important variable that we want to look at is variance. This is going to be a measure of the axial mixing within the extruder, and this variance allows us to see how wide the tracer distribution is for the mean residence time. This data is important because if we have heat-sensitive material or degradation of the API or polymer, it allows us to know what our target residence time needs to be. For a fixed length over diameter extruder and a fixed screw design, feed rate has the largest impact on residence time and the residence time span. Decreasing the feed rate increases remastication within the extruder and therefore increases the mean residence time and span. Decreasing the process temp decreases the mean residence time due to a decrease in the polymer's modulus. However, mostly residence time is independent of viscosity, and feed rate is the main variable that we like to turn to change our residence time within the extruder. Not shown here, but there is also a minor effect of screw speed on residence time. The next part that we'd like to discuss is the initial range finding in the development scale to further define the operating space. This process is often tailored to the amount of API available for the body of work. Typically, this process involves a simple parametric study or screening design of experiments to fully explore the operating space of the extrusion process. So with this flowchart, we generally like to set it up to use the continuous variables, which are feed rate, screw speed, and barrel temperature, to initially observe whether the extrudate is translucent. So once the material comes out of the extruder, you could visually observe whether or not the material is translucent. So if this material looks translucent, we will continue with the first tier testing. The first tier testing that we like to look at are PXRD, SEM, polarized light microscopy, or hot stage microscopy, all to look at whether or not the material is amorphous. Next, we will look at DSC to determine if any phase separation has occurred. And occasionally we will use vibrational spectroscopy or solid-state NMR to look at the melt homogeneity. Some of the second tier testing that we will take a look at are dissolution performance and free drug measurement, and then also potency and purity of the dispersion. So if the polymer or API has degraded or the polymer shows signs of any browning, we will go back to the parametric study and decrease the specific energy within the system to try to manufacture the amorphous dispersion. We will decrease the specific energy by lowering the feed rate and or the screw speed of the extruder. We can also lower the barrel temperature or decrease the mixing by altering the mixing sections, kneading block design, or in extreme cases, the length over diameter of the extruder. However, if the initial extrudate is not translucent, or if the tests indicate it is not homogeneous, meaning that it is phase separated or crystalline, we will increase the specific energy by increasing the temperature or mixing in a succinct fashion that allows us to determine our operating space. The last discussion we will have is the process by which we determine the scalability or the scale-up activities for a project. So this slide shown here shows how we typically scale a process from our development scale to the pilot scale and potentially the commercial scale. For this slide, it's important to note that these rules of thumb are for the transfer from a Lystritz 18 millimeter twin screw extruder to the Lystritz 27 millimeter twin screw extruder. So we typically do the scale-up process by looking at three different numbers: the power number, the pressure number, and the throughput number. For the power number, for equal viscosities and equal length over diameter at this scale, comparing the power to volume ratios allows us to equate the power number to knowing that we need to only maintain a constant screw speed between these scales, between the 18 and 27 millimeter. For the pressure number, again, for equal pressure numbers at two scales and for equal viscosities and length over diameters for two scales, all we need to do is match the screw speeds. And finally, for the throughput number, we base this calculation on the volume of the extruder. The ratio of volumes of the extruders leads to a 3.5x increase in throughput from the 18 millimeter to the 27 millimeter. So by transferring the process from an 18 millimeter to a 27 millimeter, our general rule of thumb is that we will allow a 3.5 times increase in the mass throughput through the extruder. It is also important to note that scaling between extruders of different manufacturers, you will have to look at a variety of different variables to ensure that the extrudate qualitatively is the same between the extruders. The various different things that you'll want to look at are specific energy input into the system, mixing rates, and residence time of the material within the extruder.
In summary, amorphous solid dispersions are widely applicable, and hot melt extrusion has precedence in the market. Hot melt extrusion can be screened using material sparing methods prior to large batch processing on an extruder. There are many considerations for processing techniques, and the operating space for hot melt extrusions is well defined. Dosage form solutions and BRIM research have extensive experience in addressing DCS Class II compounds, and we appreciate your attendance.
Thank you very much, Charlie and Scott. We're now going to go to our Q&A round. So audience members, if you have any questions for Charlie or Scott on today's webinar, then please submit them via the Q&A widget. Okay, so the first question of today is, what polymer is the most commonly used polymer? What are the typical advantages?
Probably the most typical polymer used for hot melt extrusion is PVP. Reasons for using PVP include the melting temperature being advantageous for formulating, as well as amendable milling properties of dispersions manufactured with this polymer. There are also a handful of polymers on the market today that are relatively new and new generation polymers that are being evaluated. However, historically, PVP is the most common polymer for HME processing.
Okay, thank you very much. And the next question is, what is the max percentage drug load in a formulation for this technology?
The maximum drug load for an HME formulation or an amorphous solid dispersion formulation can vary widely and is very dependent on the physicochemical properties of the ingoing API. Formulations can be kinetically stabilized at 50% drug loading and even higher. However, as mentioned previously, this is very dependent on the physicochemical properties of the compound and the propensity of that compound to crystallize and phase separate.
Okay, thank you very much. The next question is there a technique to easily screen HME feasibility with limited material?
Yes, there is. As many of you are aware, it does require a significant amount of material to run a feasibility batch on an extruder. There are a couple of methods to do this with very limited material and material-sparing techniques. One of those methods is spray-dried dispersions. Because spray drying can be done on a very small scale using very limited material, feasibility of the polymer as well as the drug loading can be evaluated using spray-dried dispersions to evaluate performance as well as predicted physical stability. Once the feasibility, once the feasibility is shown using spray-dried dispersions, the process can then be transferred onto an extruder. An additional technique that can be utilized for screening HME feasibility is a modulated differential scanning calorimetry. Using a DSC, a sample can be prepared with a polymer and an API, heated to a given processing temperature, and held for a various amount of time. After doing this, the formulation can be evaluated for performance, chemical stability, as well as homogeneity. However, it should be noted that the lack of mixing in a DSC experiment does make this a worst-case scenario. This technique can also be used to screen various loadings as well as various polymers. The one aspect of HME formulations that is not evaluated using these two small-scale techniques is the ability to mill the material once it is extruded.
Okay, thank you very much. The next question is, why is it recommended to use TiO2 for residence time?
We generally use titanium dioxide for this test because that's essentially the method that we have developed. We generally use it for a placebo manufacture to determine the residence time distribution at the set parameters that we have for the operating space. For the example that we listed in this process, the Raman spectrophotometer allows us to take a look at the PAT that we're testing with basically titanium dioxide.
Okay, thank you very much. The next question in the Q&A is, what is the preferred extruder size for feasibility?
The preferred extruder size for feasibility is going to be mainly dependent upon the amount of active material available. If we have enough material that allows us to look at various different processing conditions to define the operating space, we generally like to start at the 18 millimeter extruder. The wide operating space of this piece of equipment allows us to produce fairly small quantities of material up to fairly large quantities of material, all on the same piece of equipment, while defining a fairly broad operating space. So if we don't have a large amount of active available, we can do small scale screening, as Scott had mentioned, on the spray drying side of things with a mini spray dryer, but we can also use the 16 millimeter extruder to start to explore the formulation and process operating space.
Okay, thank you very much. The next question is, how much API is needed for initial feasibility?
Using some of the material sparing methods mentioned previously, very limited amount of API can be used. Anywhere from 10 to 100 megs of formulation can be used for the material sparing evaluation and HME feasibility. Once transferred onto an extruder,
then generally for the 16 millimeter, we like to say that generally anywhere from 10 grams up to 100 grams of active, depending upon your active loading in your formulation. So anywhere from 10 to 100 grams on the 16 millimeter up to, on the 18 millimeter, we generally like to have 100 to 500 grams of material, of active drug, sorry.
Okay, thank you very much. The next question is, what is the most common way to manufacture a tertiary formulation?
So if a tertiary system is required, oftentimes, you'll have your binary system of active and polymer. And so the tertiary system becomes relevant when a plasticizer or a dissolution aid is required to be added to the formulation. The addition of this material is often dependent on whether or not the material is a liquid or a solid. If the plasticizer or dissolution aid is a powder, oftentimes we can add this material to the physical blend of the active drug and polymer. We can also add the dry powder downstream of the feeding zone of the extruder in a separate block of the extruder. If the plasticizer or dissolution aid is a liquid, the extruder contains liquid injection ports that can be utilized, and this material is added to the extruder shortly into the mixing zones to ensure that the material is rapidly mixed into the active and drug system.
Thank you very much. The next question is, could you please go over the rule of thumb for the relation T to Tg in relation to stability again?
Sure. So the relationship of Tg minus T is a mobility parameter. And what we use as a rule of thumb is that storing a kinetically stabilized formulation at least 20 degrees below the Tg will help ensure good long-term physical stability. So, for instance, if the glass transition temperature of your formulation at 75% RH is 45 degrees Celsius, we would not recommend storing that any higher than 25 degrees Celsius. The way we evaluate formulations is using ICH stability conditions as a guideline. So for formulations, when stored at 40 degrees Celsius and 75% RH, we prefer those formulations to have a glass transition temperature of at least 65 degrees Celsius at 75% RH. If that glass transition temperature is not achievable for a given system, one way to mitigate that is to package and protect against humidity. So for a formulation with a low glass transition temperature at high humidity, we can package the sample with desiccant to limit the amount of relative humidity and maintain a glass transition temperature that will ensure low mobility and good predicted physical stability.
Okay, and now for the final question of today's webinar, and it is, what is the maximum API load that can be successfully handled in HME platform?
So as mentioned previously, for solid amorphous dispersions in general, this question is very compound dependent. The one limitation of HME for amorphous solid dispersions is that the active formulation must be miscible in the polymer, unless that active formulation, well, it must be miscible in the polymer, and that itself can have limitations as well. So the answer to the question would be it is very compound dependent for amorphous solid dispersions and even more so for hot melt extrusion amorphous solid dispersions.
Okay, thank you very much, Charlie and Scott, for answering all those questions in the Q&A round. If we haven't answered your question, hopefully we'll be able to, Charlie or Scott will be able to get back to you. So that just leaves me to thank Charlie and Scott. And would you like to leave us with a comment today?
No, we'd like to thank everybody for joining us. And again, if you have any questions, please use the email that we have provided on the last slide, and we'll be sure to respond when we get a chance.
Thank you very much, Charlie and Scott, for what was a great presentation, and thanks to Informatica for sponsoring this session. To the attendees, you will receive an email shortly telling you how you can access the on-demand version of this webinar, or you can access this through our website, which is www.business-review-webinars.com. We look forward to sharing further webinars with you, so please keep an eye out on our website and follow us on Twitter at BRWebinars for daily updates, and join our LinkedIn group Business Review Webinars. Thank you once again, and I hope you all have a nice day or evening.